Good day, this is the Professor, CBMD PhD, and today's Sunday, October 22nd, 2023. It is 4:39 and 41 seconds, AM Eastern Standard Time, and today's research, mitochondrial protein and cancer. So let's jump to it here. Mitochondrial protein plays key role in glioblastoma and therapeutic resistance. Date October 17, 2023. Source: Institute for Genomic Biology, University of Illinois. Glioblastoma is the most common type of brain tumor that affects adults and unfortunately still remains incurable. In a new study, researchers have demonstrated that a specific mitochondrial protein plays an important role in glioblastoma and can therefore be used as a potential target to reduce tumors. Glioblastoma is notorious for its lethality. One of the major challenges is that it spreads invasively throughout the brain. They are interested in understanding what drives this process in order to identify new therapeutic strategies. In the current study, the researchers focused on the mitochondrial coil helix, domain containing protein 2, also known as CHC-HD2. The complicated name refers to the structure of the protein, whose subunits are coiled together like rope strands. The researchers first looked at the Cancer Genome Atlas glioblastoma database to see whether they could spot any patterns that related CHC-HD2 levels to cancer. Out of 577 samples, they found that the CHC-HD2 genes had higher expression in tumor cells compared to non-tumor tissue and was higher in advanced cases of glioblastoma. They also learned that in humans, the gene encoding CHC-HD2 was closely linked to the gene encoding the epidermal growth factor receptor, or EGFR, on chromosome 7, a mutated version of the protein that's found in over 50% of the glioblastoma patients. When the results of the database showed that CHC-HD2 expression was highest in the patients who harbored the mutation known as EGFR8, the researchers realized that understanding the interaction between these two proteins can be crucial to understanding glioblastoma progression. And to confirm their hypothesis, the researchers looked at the effects of CHC-HD2 on tumor growth in mice. They compared mice that had CHC-HD2 and the mutated version of EGFR to mice that did not contain CHC-HD2 but still had mutated EGFR. The first group of mice survived for an average of 17 days, whereas the second group survived for 25 days. The researchers found that the mice that died earlier had more tumor growth infiltrating the surrounding brain tissue. Although it is evident that these proteins are interacting with each other, it is unclear how they affect glioblastoma progression. The authors have proposed several mechanisms based on their experiments. The first possibility is that CHC-HD2 decreases the sensitivity of mutated EGFR to cytotoxic drugs. Patients with glioblastoma only survive for 15 to 20 months, despite being subjected to an arsenal of treatment procedures, including chemotherapy with the drug temozolomide. The researchers used this drug and tested cells that had the mutated EGFR protein and CHC-HD2 and cells that only had EGFR. They saw that when cells did not contain CHC-HD2, when they were more sensitive to temozolomide, the researchers believe this result highlights CHC-HD2 as a potential therapeutic target. Another possibility is that CHC-HD2 affects how EGFR stimulates glioblastoma invasion using a hydrogel to replicate the tumor microenvironment in the brain. The researchers showed that the cells with both CHC-HD2 and mutated EGFR were able to grow and invade the surrounding area. And this invasion was especially pronounced when the level of oxygen was lower, a condition commonly experienced by glioblastoma cells as they invade from the tumor into the brain. The data also demonstrate that CHC-HD2 profoundly affects cellular metabolism, which the researchers showed using different biochemical tests. All of the data are outlined, and they can pursue any one of these mechanisms to see if it explains what is happening in the tumors. But researchers are interested in figuring out why the invasive capacity of these cells change, especially under low oxygen conditions. They also want to test whether these results hold true in other types of glioblastomas, which may not have a mutated EGFR. And this study suggests that the role of CHC, HD2, in glioblastoma progression was underappreciated, but there might be a significant new target as they try to improve clinical outcomes. The idea that this protein can change behavior based on its environment is valuable. They hope that researchers can start identifying new therapeutic strategies based on this key protein. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.